Hello everyone and welcome to our second lecture. Today we're going to be talking about amino acids and proteins. So I am starting with this slide that we actually saw yesterday, which again I've added just to emphasize the importance of the central dogma, which is that information starts in DNA and that's where it's normally stored in the cell. Then when a certain gene is to be expressed, the messenger RNA is created. DNA and RNA, as we've discussed, are both types of nucleic acid, which are made up of monomers, which are similar. And then the proteins are translated from the messenger RNA. Proteins are strings of amino acids, so those proteins are the polymers. The monomers are amino acids. Another name for proteins is a peptide chain or a polypeptide. So again, we discussed all of this yesterday and how important it is that these three different types of molecules and these stages in the cell are linked and how important that linkage is. And we'll keep coming back to this for the rest of the class, really. Okay. So we also briefly discussed some of the small molecules in the cell. And so in this slide, again, we have a list of the important small molecules, which include amino acids, of which we have 20, at least as humans and animals, we have 20 different amino acids. For aromatic bases, which are the nucleotides, we have 5. Remember, we have 5 because we have one base that differs between RNA and DNA, and we'll talk about that tomorrow. And then we have a number of different types of sugars in the cell and different lipids, and we won't get into that right now at all. So this is a chart showing the identity of all the different amino acids. And then we have some common sugars as well as common lipids in the cell, and on the far right we can see that we have the function of all these small molecules. So again, I think that this is a really good chart to look over for reference, especially knowing the functions of these small molecules and being able to recognize them, not necessarily write the full list of the 20 amino acids. So in order to talk about proteins, we do have to talk about the monomers, which are amino acids, and we need to talk about the chemical structure of these amino acids. So what we see here is a simple depiction of an amino acid. All of the 20 amino acids have some structure in common. Actually, most of their structure is shared between the 20 different amino acids. What I mean by that is that every single amino acid has this central carbon. Attached to the central carbon is one hydrogen, and then the other chemical groups that can be attached to the central carbon are an amino group, which is where this molecule gets its name, and a carboxyl group. Finally, at the bottom here, we have something called an R group. Another name for this is a functional group. So let's write that over here. So an R group is the same thing as a functional group. And that's the same thing as a side chain. In terms of amino acids. So again, those three names mean the same thing. They all refer to this R. So why is there an R here? And what does it mean that it's in red? And also all the other groups here are actually made up of atoms, whereas the R does not represent an atom. Well, this R is a representation of a chemical group that changes depending on which amino acid you're talking about. So for the simplest amino acid, which we see here on the right side of the screen, this is glycine. The R group for glycine is a hydrogen. So if we were going to say, all right, this is on the left here, we're going to make this into glycine, this is what it would have in terms of its R group. It would just be a hydrogen. So we end up, if we're looking at glycine, having two hydrogens coming off of the central carbon atom. And that is what we see on the ball and stick model here on the right. So this gray atom is the central carbon. It has the two hydrogens coming off of it. One of them is the normal hydrogen that every single amino acid has. And then the other hydrogen on glycine in particular is the R group, because the R group of glycine is a hydrogen atom. Now, we also see the amino group, and on the other side, a carboxyl group. Remember that both of these are also attached to the central carbon. Now, the central carbon is important because it is always going to be the atom that has 4 bonds, one to the amino group, one to the carboxyl group, one to the hydrogen, and then one to the R group. And that is consistent between all of the amino acids. The only thing that changes between the identity of the different amino acids are the R groups. In addition, when we start to see the formation of polymers of a string of amino acids, we're going to see some changes in the amino group and the carboxyl group in order for these guys to form bonds between the multiple amino acids. So something else that I wanted to mention, and this just details what I just said,